Hello friends, I am Dr. Nitish Bhatia and I am here for some mass evolution classes. Welcome to our YouTube channel. In this video, I will explain the concept of pharmacokinetics and its importance. In this presentation, I will tell you about pharmacokinetics. Pharmacokinetics is the study of disposition of a drug. The disposition of a drug includes the processes of A, D, M and E. That means absorption, distribution, metabolism and excretion. Metabolism and excretion are collectively known as elimination. Apart from this, I will be telling you something about toxicity of drugs. Metabolism and excretion, as I have told you, they are collectively known as the elimination of drugs. Now, here is a diagram of the fate of a drug after oral administration. We know that the movement of a drug substance from gastric or intestinal lumen to the systemic circulation is called the process of drug absorption. Now, the absorbed drug content then undergoes distribution in the systemic circulation, and while doing so, some proportion of the drug content gets bound to plasma proteins and bodily tissues, depending upon the physicochemical characteristics of the drug itself. Moreover, the drug molecule also undergoes the process of metabolism. Now, metabolism is a collective term for a series of chemical reactions that are intended to render the drug molecule inactive or ionized in order to aid the excretion of the drug through renal or biliary excretion phenomena. Now, let us take each process, each phase into more details. First of all, the process by which a drug proceeds from the site of its administration to the site of measurement, that means the bloodstream, the blood plasma within the body, is called absorption of the drug. Absorption is necessary for the production of therapeutic effect. Most drugs, they undergo gastrointestinal absorption. The extent to which the drug is absorbed from the gut lumen into the portal circulation is an important pharmacokinetic parameter called the bioavailability. IV or intravenous administration has 100% bioavailability and is considered as a standard. Moving on, the process of absorption relies on many factors. Some of them are: number 1, the passage of drug molecule through the cellular membranes to reach the blood circulation or plasma. Number 2, passive diffusion in case of lipid soluble drug species. Pharmaceutical scientists all over the world always remember the rule of 5 in determining the formulation characteristics of a drug. Poor absorption or permeation is more likely when, number 1, there are more than 5 hydrogen bond donors in the structure of a drug molecule. Number 2, the molecular weight of a drug is over 500. The log p value is over 5 and there are more than 10, 5 into 2, 10 hydrogen bond acceptors in a drug structure. We should always remember that non-ionized or lipid soluble molecules can only diffuse across the cell membrane easily. It is very difficult for the ionized molecules to cross the cell membrane. Now, let us talk about an important aspect of pharmacokinetics, that is first pass metabolism. A drug molecule faces many barriers in its journey from the site of absorption in order to reach the bloodstream or systemic circulation. It may get destroyed by the gut acid content, or it may get destroyed by the gut wall enzyme before it even reaches the plasma, the blood plasma, or before it gets absorbed. Even after absorption, the hepatic portal vein first takes the absorbed drug to the liver, where it gets metabolized into inactive and sometimes active metabolites. So, we talked about bioavailability in our previous slides. In order to estimate or determine the bioavailability, pharmaceutical scientists consider absolute bioavailability as a standard. A drug molecule, a drug given by IV route will have an absolute bioavailability of 1 or F is equal to 1, 100% bioavailable drug. While drugs given by other routes usually have an absolute bioavailability of less than 1. So, we consider 1 as standard, other routes less than 1. The absolute bioavailability is calculated by the area under curve non-IV divided by IUC of intravenous. Now, let us talk about next pharmacokinetic parameter, that is distribution. Distribution is the movement of the drug from the blood or plasma to all tissues of the body. This means movement from the blood to interstitial fluid to intracellular fluid and back and forth. Distribution is determined by partitioning across various membranes of the body and binding of tissue components, binding of blood components. Now, blood components including plasma protein, RBCs, as well as physiological volumes or volume of distribution. All of the fluid in the body, which is also referred to as total body water, all of that fluid in which the drug can be dissolved can be roughly divided into 3 compartments. Total body water can be divided into 3 compartments. Number 1, intravascular, blood plasma found within the blood vessels. Number 2, interstitial water, interstitial tissue fluid, fluid surrounding the cells. Number 3, intracellular fluid, fluid within the cells, which we also call cytosol. The distribution of a drug into these 3 compartments is dictated by number 1 its physical, number 2 its chemical properties or physicochemical properties. Now, another important parameter is apparent volume of distribution or VD. VD is equal to the amount of the drug in the body divided by drug plasma concentration. Volume of distribution of some drug species can vary. For example, cocaine has a volume of distribution of 140, clonazepam has a volume of distribution of 210 liters, amitriptyline has a volume of distribution of 1050 liter, and amiodarone has a volume of distribution of 5000 liters. Now, coming on to factors affecting volume of distribution. Number 1, the blood flow. Its rate varies widely as a function of the tissue. For example, muscle have a slow blood flow rate when we are at rest. Organs such as lungs, liver have fast blood flow rate. Second factor is capillary structure. Most capillaries are leaky and do not impede diffusion of the drug, but the capillaries of the blood brain barrier, which are formed by high level of tight junctions between the cells, they are not leaky, they are almost impermeable. So, blood brain barrier is almost impermeable to most of the water soluble drugs. So, blood brain barrier can be permeated by pharmaceutical scientists for pharmaceutical purposes by number 1, distribution of osmotic means, disruption of this barrier by osmotic means, use of endogenous transport systems blocking the active reflux of transporters present at the blood brain barrier cells, or by intracellular implantation. Moving on to next factor, plasma protein binding. So, many drugs bind to plasma proteins in the bloodstream. Plasma protein binding limits the distribution of the drug. A drug that binds plasma protein diffuses less efficiently than a drug which does not binds. Now, coming on to next parameter, partition coefficient. The partition coefficient or POW can be used to determine where a drug is likely to go in the body. Any drug having a partition coefficient greater than 1 will diffuse through the cell membrane easily and is likely to be found throughout all the 3 body fluid compartments. Drugs with low partition coefficient value, meaning that they are fairly water soluble, are often unable to cross the cell membrane and they require more time and body fluid to distribute throughout the rest of the body. Now, molecular size of a drug is also another important factor in pharmacokinetics. The size of a drug also dictates where it can go into the body. Most of the pharmaceutical drugs, their size ranges between 250 to 450 daltons. tiny drug molecules, small molecular size drug molecule 150 to 200 Daltons are having low partition coefficient value like caffeine, which can passively diffuse through the cell membrane. Antibodies and other drug molecules can range into thousands of Daltons. Drugs having more than 200 Dalton mass value cannot passively cross cell membrane, and therefore, they require specialized protein based transmembrane transport systems, which limits their distribution. Drugs having less than 1000 Dalton weight with high partition coefficient simply diffuse between the lipid molecules that make up these membranes, while anything larger than 1000 Daltons requires specialized transport systems which are present inside our cells. Now, let us talk about the process of elimination of the drug. Now, elimination is the irreversible removal of the parent drug from the body. Elimination takes place by metabolism as well as excretion. These two processes combined known as elimination. So, let us first talk about the drug metabolism. It is a chemical modification of the drug, that means its molecular structure, with an overall goal of getting rid of the drug. Enzymes of metabolism which are typically involved in metabolism are cytochrome P450 enzymes. The aim of metabolism is to make the drug more polar or more water soluble, so that it can be excreted by the renal excretion. Now, let me tell you something very interesting. This is regarding a drug which is highly abused, heroin. From 1898 through 1910, you will be surprised to know that heroin was marketed as a non-addictive morphine substitute as a medicine for cough for children. Bayer Pharmaceutical marketed heroin as a cure for morphine addiction. It is very interesting to note that in our body, heroin gets converted into morphine when it is metabolized in the liver. So, you can very well imagine something which is used for the treatment getting converted into the drug of abuse only. Now, let us talk about the phases of drug metabolism. Phase 1 reactions. Phase 1 reactions, they aim to convert the parent drug compound or molecule into a more polar or hydrophilic metabolite by adding an unmasking functional group such as hydroxyl group, thiol group, nitro group, example oxidation, nitrosation, sulfation, so on. Often these metabolites are inactive or are readily excretable, or these molecules may be sufficiently polar to be excreted readily by the body. Now, let us talk about phase 2 reactions. These reactions involve conjugation of the drug molecule with an endogenous substrate to further increase the aqueous solubility of the molecule. For example, conjugation with glucuronide, conjugation with sulfate, acetate, amino acids such as glycine conjugation is very important. Metabolism, which is also known as biotransformation, mostly occurs in the liver cells or liver, because all of the blood in the body, all of the drug which comes into the blood, it first passes through the liver by hepatic portal system. The most important enzymes of metabolism phase 1 are cytochrome P450 enzyme or microsomal cytochrome P450 monooxygenase family of enzymes. Now, these enzymes tend to oxidize the drug molecule. These enzymes act on the structurally unrelated drug molecules. These enzymes metabolize a wide range of drugs. Cytochrome P450 enzymes are very special as they are found in liver, small intestine, lungs, kidney and even placenta. They consist of more than 50 isoform or subfamilies of enzymes. They are the major sources of catalytic activity for drug oxidation. It has been estimated that 90% or more of the human drug oxidation can be attributed to only 6 isoforms of cytochrome P450 enzyme: CYP1A2, CYP2C9, CYP2D6, CYP2E1, CYP2C19, CYP3A4. It is interesting to note that in different people and different population all across the globe, activity of different cytochrome P450 enzyme oxidases differs. Inhibitors and inducers of microsomal enzymes. Certain drugs can act as inhibitor, for example, cimetidine, which prolongs the action of drugs by inhibiting cytochrome P450 enzymes. There are certain drugs which are inducers of cytochrome P450 enzymes, drugs such as barbiturates. carbamazepine. They shorten the action of drugs, increase the effect of those biotransformed into active agents. There are certain drugs which are blockers of cytochrome P450 enzyme, acting on the non-microsomal enzymes such as MAO inhibitors, anticholinesterases. Now, let us discuss more about the phase 2 metabolism reactions or conjugation reactions. Main function of phase 1 reaction is to prepare a chemical for phase 2 reactions. Phase 2 is the true detoxification step in the metabolism process. Detoxification means making a molecule less toxic or non-toxic. Phase 2 reactions are also known as conjugation reaction, which include, as I have told you, glucuronidation, sulfation, oxidation, all these reactions. Acetylation is also a type of phase 2 reaction. Amino acid conjugation with the help of a very important amino acid such as glycine is also a type of phase 2 reaction. An important molecule, glutathione, conjugates to major drugs and which undergoes phase 2 conjugation reactions. Fatty acids are also used in conjugation. Even condensation reactions are also involved in phase 2 reactions. So, overall, if we summarize phase 1 and phase 2 reactions, we can say that the product of these reactions are generally more water soluble. These reaction products are ready for renal excretion, and there are many complementary, sequential and competing pathway involved in these reactions. Phase 1 and phase 2 metabolism are a coupled interactive system in our body interfacing with endogenous metabolic pathway in order to render drugs less toxic and easily excretable. Let us discuss the last pharmacokinetic process of elimination, the main process that body uses to eliminate the unwanted substances out of the body. Most common route of elimination is biliary or renal elimination or excretion. Other routes involve lungs through exhalation, skin through perspiration. Lipophilic drugs may require several metabolic steps before they are excreted out of the body. Now, let us talk a few things about the toxicity. So, toxicity is the noxious or detrimental effect of a drug molecule produced at the doses higher than the therapeutic doses. So, whenever we are taking a drug at higher than the therapeutic dose, it produces toxicity. The therapeutic index is the degree of separation between the toxic and the therapeutic doses. Relationship between the dose, therapeutic effect and toxic effect can be seen in the study of toxicity. The therapeutic index is narrow for most of the anti-cancer drugs. The more the therapeutic index is, the more safe the drug is. So, in last, I can summarize that the process of absorption followed by distribution of a drug molecule are responsible for drug action, whereas the process of metabolism and excretion taking place simultaneously are responsible for the inactivation or stoppage of drug action. All these processes are studied in the branch of pharmaceutical sciences known as pharmacokinetics. Simply what the body does to the drug. I hope you have enjoyed this video. If you have enjoyed this video, please do like and subscribe our channel Soma's Evolution Classes, and for more interesting videos, stay tuned with our channel. Thank you.